Hello and welcome everyone to this video podcast for the Biology 211 lab. Last week we focused on how we went about isolating DNA, amplifying it using the technique PCR, and analyzing that using gel electrophoresis. As is true with any lab experience, a large part of a project involves writing, and thus one of the most important skills a scientist has is that of writing. Your final project for this first module is to write an introduction. So today we're going to spend some time setting up how to best approach writing that introduction, and that is by generating an outline. So that's going to be the first part of today's material, and that is to complete the worksheet in your worksheet packet that involves writing an outline for your introduction. You will also create and annotate figure 1. Now this will be the image from the initial PCR. You'll use the image I have posted on Blackboard to construct this figure. You'll also learn how to write an effective legend that goes along with the figure. All that is due this week on Thursday is the introduction outline. The figure that you complete will be due next Tuesday. Next Tuesday, in fact, you will have 2 figures and the introduction due. However, by the end of this week, you should have all of that done, and you could even submit that early if you wanted to. Now remember, this is the first time we've taught Biology 211 online. And so it's the first time we've done the lab online. And as I generated the schedule for the lab, I knew there would be some tweaks that we would have to do. And this week there's going to be a small tweak. I think in the end it'll generate more free time for you next week, but I think it'll flow a little bit better. So what we're doing today, as I've already told you, is we're going to work on the outline and then make figure 1 with its legend. And then tomorrow we're going to understand how we set up a sequencing reaction, what that entails, and how it's similar to replication, but not exactly like replication. Then you'll make figure 2 and start writing the introductions. In fact, you could even start writing the introduction today, right after you've completed the outline. Then on Thursday, you're going to analyze the sequencing results, and I've posted on Blackboard the sequencing results from Arabidopsis, kale, and cabbage. And then you'll peer edit each other's introductions and their figures. So what that means is, before you come to class, or before you do this activity, you have to be able to have a rough draft of your introduction. It doesn't have to be perfect. In fact, it probably won't be perfect. It'll give you a chance to get some feedback from your group. The part that I changed was, initially I had analyzed sequences, this part here, which is really lab 1.5. I had that on during week 3 on Tuesday. But I thought if I just moved it here, because in all honesty, it'll take you maybe 10 or 15 minutes to do. It's a very short thing to do. And that way, your whole Tuesday of next week is opened up. There's nothing specific for that day, except it will give you time to complete your introduction and figures. Or if you get that done, the introduction and figures, during week 2, then there's really nothing that's going to be due that day, or that you're responsible for that day. It'll give you a chance to get caught up on other things or to prepare for the next lab. Before we get started, I think it's important for you to understand some of the history behind how we developed these courses, the foundational courses. And one thing that we wanted to do was break up how to write a lab report. In our previous curriculum, the introductory course had you write an entire laboratory report your very first semester of your freshman year. We realized that was probably asking way too much of anybody to do that the first semester. So what we've done is broken up these different steps so you can learn and refine them throughout the 5 foundational courses. You'll see in Biology 211, we will write a hypothesis, and that will come in the lab report for the next module. That's a repeat from 111, so we're going to build upon that. We're going to build upon the methods in our lab report for Module 2, as well as the results and the discussions, which you first did in Biology 112. This will be the first time you've done an introduction, because that was not in 111 or 112. That is, unless you've already taken Biology 213, which is possible. And an outline. This is the only course that really spends a little bit of time talking about how to effectively make an outline. Just to give you a little bit of an idea how the writing builds up in each of the courses and how it's fortified in other courses. So let's talk about an introduction in a little bit more detail by talking about some key features of an introduction. Now remember, the introduction is going to be the first thing that is read. So it has to set the stage for your entire lab report. Or more likely, it's going to set the stage for your scientific argument. Often in science, we build up evidence in the introduction to lead the reader to the next question that we feel needs to be addressed. And then that is what the rest of the paper is about. The introduction also needs to hook the reader. They should see why the study is interesting and why it is significant. And this is one of the more important things that an introduction does. It has to summarize the literature that's already been published that highlights the questions that you're addressing in your paper, explains the rationale for the study and the major objectives. I often think of it as providing a summary of that literature, and it should lead the reader to the next most logical question that has to be asked, or thinking about it as identifying the hole in the literature. What hasn't yet been addressed yet? And that's why we need to do this study. Now, please read Chapter 4 on introductions in your Macmillan book and pages 33 to 34 in your lab manual. This will give you some highlights of what a good introduction should have beyond what I'm telling you here in this PowerPoint. Work on the introduction outline worksheets, remembering to include the references that you find and use. Any references you identify today while you're working on your outline, go ahead and write those down so you don't forget them. In scientific writing, we don't use direct quotes. So in your introduction, you should not use direct quotes. We always use our own words to describe previous findings in the literature. Now remember, the completed worksheet is due Thursday at 11:59 p.m. As you're writing your outline, I'm going to provide a few tips for you. To begin, I'd recommend finding and reading sources relevant to the research project you're working on. We've provided 2 of these resources for you on Blackboard. I would make sure to include those in your outline and in your introduction. Now, it's perfectly okay within your groups to work together to find additional information, but make sure to complete the writing in your own words. Make sure to use all the sources of information you have. You have your lab manual. There's a lot of important background information in that manual. In addition, there are highlighted references in that manual that you might want to go to. The lab worksheets and the pre-lab PowerPoint slides. The 2 references I mentioned that are on Blackboard. There's a thesis by Maggioni and another paper by Schrantz and Mitchell-Olds. As I said, there are several papers referenced in Lab 1.1 that have significance for GAPDH, and you also might want to just Google the authors' names and you might find other papers by them. There are many different types of writing styles. In reality, in science, we change the writing style from one paper to the next depending on which journal we're submitting our paper to. But to be consistent throughout the biology foundational courses, we're going to use the CSE style because that's the one that's found in your writing manual. Make sure you review Chapter 6 in Macmillan. That's pages 132 to 141, depending upon which edition you have. So they might be off by a page or 2. You'll also want to look in Appendix 3 of your lab manual, and that's a quick guide to the CSE formatting. That should be on page 161. Now, in addition to writing the outline today, I also need you to design a figure and write the legend. But first, let's think about the figure. Now let's take a peek at this uncropped gel. It has 6 lanes, and this isn't a gel of what we would have seen in our initial or nested PCRs. This is just a random gel. But in this particular gel, you have molecular weight ladders in lanes 1 and 6. 4 and 5 are 2 negative controls, and 2 and 3 are the test samples that were ran. Now, when you have a figure like this, you want to make it as presentable as possible without altering the data. One way we do that is by cropping off the parts of the image that don't have any data in them. So let's back up here. These large areas here that don't have any DNA that was ran in them, we can just cut those off. You can also cut off at the top of the gel as well, but you can't cut off where the wells are. That's part of the data, so to speak, because it's important for us to know where the DNA started in the gel. You can resize the image from a corner, but never stretch it from side to side. Depending on which program you have, just make sure that when you're enlarging your image, that you're not distorting it. It needs to enlarge in an equal way. So after you've done the cropping, this might be what that figure looks like. You have 6 lanes, as I said in the previous slide. You want to label those lanes simply with just a 1, 2, 3, 4, 5, and 6. That's what you'll want to do on your gel images as well. Label what the samples are in each of the lanes. One of the keys to making a good figure is that a reader should be able to look at that figure and the legend and be able to gain all the information they need to from it easily. As a reader's looking at this, they may not know what these size markers are. In fact, it's unlikely they would off the top of their head. So we tell them, we say this is where the relevant markers are. Now, any acronyms or abbreviations you use, you're going to want to make sure you define in the legend later on. A reader doesn't have to wonder what MWM means. They can just look at the legend and be able to tell what it means. Now, let's think about writing a figure legend. Read Chapter 3 using tables and figures in your Macmillan book. When writing a figure legend, be sure to number figures and tables consecutively as they appear in the paper. Every figure or table that is present in a paper has to be referred to in the writing of the paper. You can't just have figure 2 and then never mention figure 2 in the actual text of the paper. The figures and tables should be in numerical order as they are mentioned in the text. You shouldn't talk about figure 5 before you talk about figure 2, for instance. A figure or a table, as I said, I think on the previous slide, needs to be understandable apart from the text. In general, you should be able to read any figure in a paper and understand the main points of that figure without reading the text. The image figure and the legend needs to be on the same page. So here's an example of a figure and its legend from the writing manual. You can see here that the word figure and the number 3.15, shown here, are in bold and followed by a period. I want you to use that same format. The figure title immediately follows figure 3.15. And our method will be not to bold that. And this is one of the most important parts, at least in my opinion. Everybody has their own thing that they think is the most important part, but this is really one of the most important parts, I believe. And that is that this very first sentence, the title sentence, is the summary of that figure. If a reader only read that, they would understand the main take-home point of that figure. Then they can read the rest of it to understand some more of the details of that figure. And then after that, we have additional information. So let's talk a little bit more about writing this figure legend. Choosing the right words to convey your meaning is very important. This is true in all walks of our lives, but it's also true in writing figure legends. Using the right words. The figure title needs to be in an active voice. It is not supposed to be passive. To remind you, in passive voice, the subject of the sentence receives the action. So lizards, that being the subjects, were collected from 3 different sites. You can go ahead and pause now and think to yourself about how you could rewrite that sentence so it's more active. In the active voice, the subject performs the action. We collected lizards from 3 different sites. In this sentence, we are actively collecting the lizards. As I said on the last slide, the title must be the main conclusion of the figure. We often call this the take-home message. It emphasizes the main idea that you want the reader to understand and remember. Use specific, informative language. We don't want the language to be vague. So we wouldn't say several pits were dug at each forest site and soil samples were collected from 3 different depths in each pit. Quite a bit of vagueness in this sentence. So a better way to write this sentence to provide enough information to the reader is to say 4 randomly located pits. So here, you're not saying several, you're saying 4. Were dug at each forest site and soil samples were collected from 3 depths at each pit, and it tells you specifically what each depth was. So that provides the kind of specificity the reader would want to know. So let's look at these 3 figure titles and determine what is the best take-home message about the data shown. So here's our figure. Before we even look at the titles, you can see you have an X and a Y axis, and you can see that there's this line showing live births over the age of the mother. So which one of these titles would be the best? That would deliver that take-home point. Graph of relation between live birth rate and women's age. Now this is a true statement. That's what this graph is. It shows the relation between live birth and the woman's age. Or red line shows how a woman's age affects the live birth rate. That also is an accurate statement. That is exactly what the red line is showing. Third statement here, live birth rate decreases as a function of increasing women's age. That is also a true statement. But which one of these is the take-home point? Go ahead and hit pause and see if you can think about which of these, A, B, or C, is the correct answer. The best answer is C. This is the take-home point. As I said, A and B are correct statements, but that's not really the take-home point. If you read this, live birth rate decreases as a function of increasing women's age, that tells you exactly what the authors are trying to convey. The first 2 are just kind of describing how the figure is set up. Interesting, but not a take-home point. So these are the details that you need to include in your figure and the legend. I'm not going to read this to you. You can read it, but it's also in your manual and the rubric. So look at Lab 1.4, Part 3, pages 32 and 33. This week you will construct 2 different figures. One will show initial PCR results, and the other will show nested PCR results. The figure itself is worth 20 points, and the legend is worth 30 points. Here's the rubric that you should follow when you're making these two items. If you follow all of these directions under this excellent 100%, then you should be in good shape. I'd like to spend a little time just going through an example of how I would go about making a figure. And as I go through my example, you'll see I'm going to crop and annotate the gel image in PowerPoint. I would also recommend doing the figure legend in PowerPoint as well. You could do it in Word and just copy it over, that's fine too. Make sure that figure legend appears below the figure itself. Never put it above, and never write on top of the figure 1 or anything like that. It's just the figure and the legend right below it. You'll need to turn in your figures with your introductions by Tuesday at 11:59 p.m. of week 3. You could turn them in sooner, but at the very latest they have to be turned in by next Tuesday. As you're turning them in, if you have any problems, let me know, particularly if Blackboard tells you that the size of your document is too large and it's causing you problems. Okay, I'm going to walk through how I would go about making a figure image from the data that you see here. Now, these data aren't like the data you've seen in lab. This is just data from my lab. And so what you see here are 2 yeast cells with mitochondria that are fluorescently labeled in it. But as you can see, by looking at this, it's hard to interpret it just as it stands. So the first thing I would do is take this image here and import it into a PowerPoint slide. So I'm going to open up PowerPoint here, and the first thing I'm going to do is insert my picture of the mitochondria into this PowerPoint slide. So I'm going to go up here to insert, and I'm going to insert a picture. I'm going to insert it from a file I have on my desktop, just like so, and hit insert, and there it is. Now these are just guidelines that I'm sure you've seen before, and we can get rid of those if we need to, but we're going to keep them there for now because we might use those to set up the picture. But the first thing I'm going to do is crop. And depending on the version of PowerPoint you have, there should be a crop tool somewhere up here, and this is where mine is. But if you can't find it, just go here, search in presentations for where is crop, or wherever your help tool might be. But I'm going to click on crop. And now what I can do is just draw these arrows in, and you can see as I'm doing this, the crop function doesn't change or distort my image where the figure is itself. It's just getting rid of some of that dead space. I'm going to do something like this. And then when I'm done, I just go back up here and hit crop again, and there is my figure. I can make it bigger if I want, like so. Maybe I'll make it a little bit bigger. And if you remember when I said make sure when you're doing this you don't distort the image. And so in PowerPoint, if you just move by the corner, you don't distort the image, you can just make it smaller or larger or smaller. Now if I were to move it on the side, you can see how that really distorts the image, and now it's not an accurate representation. So I'm going to hit Control Z to send me back here. Same thing if you moved it this way. Don't ever do that. So here's my figure. Now what I want to do is label this so that someone looking at this knows what I'm trying to tell them without them having to do a whole lot of reading. And so above here, I'm going to put in here 0 millimolar furfural, which is just a chemical we use that causes cellular stress. And then I'm just going to copy that. I'm going to move it over here, and I'm going to change it from 0 to 25 millimolar. I want both sides to be about the same, right? And you can use this guideline here to move up here and make sure that those letters are about the exact same position. Now what are they looking at? Well, I'm going to put in here, I'm going to make this lettering here white so that we can see it on the black background. I'm going to put tubular mitochondria. And maybe I'm going to take an arrow up here, and you can put a shape in here to show what I'm talking about when I say tubular mitochondria. I'm also going to make that white. There's no reason to be super fancy here. We want good contrast. We want good color contrast. And then I think I'm going to put it like that. And then over here, I'm going to put fragmented mitochondria. Often what I'll do is I'll just copy things and paste them. So Control C and then Control V for copy and then paste. That way I don't have to go back through and reconstruct a lot of these things that I already made. You probably know that. So fragmented mitochondria. So I can tell by just looking at this that I'm looking at cells here, 0 millimolar furfural or 25 millimolar furfural, and I can say without any furfural the mitochondria remain tubular, but when I add this chemical they become fragmented. Now I'm going to, just because I'm kind of a fan of being kind of consistent here, since this one's on 2 lines, I'm going to make this one on 2 lines. And I'm going to go back through here and just kind of change this up a little bit. The nice thing about this, you can always go back and change things. Now, one thing I would do constantly as I'm making a figure like this is I would constantly make sure I'm hitting save so that I'm not completely done and it crashes and I realize I never saved it. Don't let that happen. All right, now let's make a figure legend. I'm going to just insert a text box down here. And remember, we want to begin with the figure number. So we might go figure 1.1. And we want that to be bold. And we want this next sentence to be the take-home point. And it might be something as simple as this. Mitochondria fragmented in the presence of furfural. Okay, well, this is good. This is a good start. But let's be more specific and say in the presence of 25 millimolar furfural. And another thing we might want to put in here, because we don't know if this is yeast, if it's human cells, what kind of cells are they? We don't know just by looking at it. So we're going to say yeast mitochondria fragmented in the presence of 25 millimolar furfural. That's our take-home point. Then we can provide some details. And I'm going to position this text box so it's just right underneath the figures. That should be symmetrical with the figure itself. So I'm going to type in some details here. Okay, so you can see what I wrote here. You don't have to read it in details, but if you can if you'd like. This just provides a lot more details so that when somebody reads the take-home point, now they can see how I came to that conclusion. All right, now I'm going to tidy this up just a little bit. I'm going to move the words just a little closer to the image. I'm going to make sure the text box and the figure box are about the same. I'm going to center this whole thing as well. And one thing to be careful of is if I select the image here and move it, it's going to move, but nothing else will. We don't want that. So if you want everything to move, select all. And the way I do that is just by Control A, and that selects everything. And now I can move everything that's connected here together. Another thing you can do is up here under Arrange. You can click on that, and then you can type in this item here called Group. And now that groups everything together. It's all together. So I can hit Delete, and it's all gone. But I can bring it back with Undo. Again, make sure you save this. And now your figure is complete. Now, what I would probably do next with this is screen capture this image here. There's different methods you can do. And then copy this into a Word document. The same Word document that your introduction is going to be. So let me go ahead and do that. Screen capture. Then I'm going to open up a new Word document. And I'm going to insert my figure that I just made. So under Insert and Word document, insert picture from my file. And I know I just saved it to my desktop, and it's the first one there. And so now my figure is complete. Again, it's been cropped. I've labeled the 2 different types of yeast. I've identified structures in it, so I've annotated it properly. My figure legend, I began with the bold figure 1.1. And then the first sentence is an active sentence, and it's the take-home point. And then I provide specific detail below for the best analysis of the figure by the reader. So let's go ahead and look towards next lab. Prepare for lab 1.4. There's these different parts 1, 2, and 3, and 4. Look in Macmillan on pages 21 through 26 on reading scientific papers and avoiding plagiarism and taking notes. Some of the questions from the quiz for this week come from this section. That is all I have for this particular PowerPoint. If you have any questions, please let us know. If not, we'll see you in lab or lecture. Bye for now.